So,
Why can't we just watch your own?
and I'm also recording.
I have it on mine. You have Netflix and YouTube and...
Again, you can't talk now.
That's it. And you have other things. No, I don't. I have games.
You are now...
You also... This is impossible.
Hi, folks. Hi, Robin. Okay, I'm just going to mute because my kids are sitting there having a fight. So give me a minute. All right, folks, can we see your lovely faces? Oh, there's a face. Hi, Clara. How are we all doing? Yeah? I can't hear you because you're on mute.
Good.
Good. All right, so I know Millie isn't going to be here. I think Andrew is going to be. Can't remember who else, a few others more. How is it all going? Have you been into school or is that not a thing?
I'm there tomorrow.
Okay, I think Millie's in today. Have you been in, Robin? No? Jazz? So are you actually, if school goes back and 6 years go back, are you going to go back? Any takers?
I'm not sure yet.
Yeah, I see. I think that's like a question that no one has asked. Are people going to go back? Fallon, are you going to go back?
Yeah, it should be.
Yeah? Jazz, are you going to come all the way down?
Yeah, it depends. Like if boarding is still kind of saying, but if boarding isn't going to be saying, then probably not.
Well, obviously boarding isn't, but like I think at the moment I was in school this morning trying to just work with Mr. Duncan to get him set up, and they're all in meetings. Like they were there for at least, I was there for 3 hours and I swear they hadn't moved, so they're trying to figure it out. But yeah, we'll see what happens. So do we think we're going to have any more? 8, and I'm one of those 8. Who's John? Do you know who John is? Hey, Zara. All right, so did you get a chance to have a look at the first little bit, just the structure of the plant and asexual? Yes, that's okay. All right, I'll start sharing my screen and we'll just get through this little part. So at about 3:30, this assignment will ping for you on Teams. This is what's in it. So what we're going to look at today is male and female nuclei development or gamete development. This is the sexual reproduction in plants. We'll have gone by the time we're finished through statements 1 to 15. I'm going to give you a little table. It's not in your book because it was just when I was looking at it, I thought this may help you when we're not together, where we summarize what's different and similar in the male and female process, because there's things that are the same and a couple of little differences. There's a couple of questions in your biology plus, and there's a 2010 question. And as I say, that will all ping and you can ask questions away, but this will ping. So what we are going to try, excuse me, why are you not now doing this for me? Okay, you go out, discard, go back in. So thank you for your patience.
Is Biology Plus our big book?
Is your blue book, isn't it? This might be the wrong. because you're not biology plus. Okay, ignore that one for now. I will take that out. It may not, because it's going to populate. Take that one out for now, because it's a different book. I just noticed that. So this will ping, and I'll remove that from it. But it's just to give you ideas of questions that they like to ask for this. So the plant reproduction, it's big enough, but the issue is students don't like it. But if you're serious at doing well at your biology, you need to have plants. It's just unfortunately a no-brainer. So look, we are going to, it's page 175 in mine. Is it 175 on yours? It says male gamete formation. So all I need is 176 or page behind. So look, in the first row, you already have this picture. So this is your pollen grain. I'm going to go backwards. So in this particular picture, it is this part is the thing that is in row 1 of your page 176. So you can see here that there's 4 chambers in it. Are you not now? So if you wanted to annotate for yourself, that is a pollen sac, is one of the chambers, and each anther will have 4 of these chambers, and they're going to just be called pollen sacs. So that's just your intro into it. Now the QR codes that are spread throughout this chapter, again, they're based on the Irish system and they're good. So any issues you have a look at these. So in your row 2, and you need to shout at me now if you're not happy. In your row 2, we're actually looking down the pollen sac. We're looking at it from the top. So that's what that circular little set of diagrams is. It's going to come in backwards, isn't it? Yes, it is. So all this is just showing you are the different layers that are in each of those pollen sacs. So the outer layer is the fibrous layer, and it's obviously, it's tough because it's fiber. We have this, it's green on the screen, but it's the next layer. It's kind of the lightish color. It's called a tapetum. And this is where you're going to have the energy is going to be provided for the cell divisions that are going to happen within here, or sorry, in this little section here. So the tapetum is the food store for that. This inside layer here is called, I'm going to change my color pen. The inside layer is called the pollen mother cells, and we'll talk about them in a minute. But this is essentially where pollen is going to be made. So these are diploid cells that are going to divide to give us some type of nucleus that is going to be used in fertilization. And the little dots that are inside here, these are the little pollen grains. So we have a protective layer, a food source, we have the diploid cells, and then finally we have the pollen grains inside. So in the box that's underneath, we're going to describe what happens to one of the cells that's in this layer here, this red layer, and how it's going to make male gametes. We don't quite get there in this step, but it's the process by which we get there. So I'll just wait for you now. So I can see all your lovely faces. We all happy? Yeah, okay. So the real important part of this is the part that's kind of row 3 and row 4. So you already have the diagrams in here. Now, usually it's the words they're looking for. It's not the diagrams, but when you see the female and the male together, the diagrams are kind of similar. There's a lot of similarity between them. So the first thing is the name of this cell. So this is the cell that's found is a pollen mother cell, and it's called a microspore mother cell. So if you can think of, in terms of humans, sperm are way, way smaller than eggs. So that's it. This has the name micro. And when we go to the girl version of this, it's a macrospore. So it's a microspore mother cell, and it undergoes meiosis. Can you guys remember when we did cell division? If a cell undergoes meiosis, how many cells do you end up with? One becomes what? Do you want to show me with your fingers as opposed to, can you remember? So if you do mitosis, one cell becomes how many cells? You remember? Yeah, it becomes 2. Thank you, Jazz. And in meiosis, one cell becomes how many? 4. Thank you, Alicia. Okay, so here you get 4 cells that are made, and they call this a tetrad of pollen. And tetra is meaning 4. But in reality, these are 4 haploid cells, and they're stuck together at this point. And what happens to them then is that they will separate out, and you end up with 4 haploid cells. So microspore mother cell, meiosis, to give this tetrad of pollen, they separate. And each of these 4 cells will go through the bottom row on page 176. So it's going to undergo mitosis, but it's the nucleus that's undergoing mitosis. So if you want to put that in, it's the nucleus that is doing the mitosis this time. We're not actually separating it into 2 cells. It is 2 nuclei we're making. So we end up with this little picture here. So we have 2 haploid nuclei, and this is our little pollen grain. So what do we have? We've got 2 nuclei, but the nuclei have names. So one of them is called a tube nucleus, and it's haploid, and one of them is called a generative nucleus, and again, this is haploid. And the only reason it's haploid is because the starter over here on the left-hand side was haploid. So the nucleus undergoes mitosis to give me 2 nuclei, and now my pollen grain has what's called an extene and an intene, and I'll explain what they are in a second. So microspore mother cell, meiosis, tetrad of pollen, they separate. I have a haploid cell undergoes mitosis to give me 2 nuclei. And the names are important. So tube and generative. So we have Hannah, we have Robin, we have Zara, no Grace, no Peter, no Shayna. We have Flo, Velen, no Concave, Clara, no Jackson, no Tig. I didn't see Laura, I did see Laura. I have no Jess, I have Mark, I have Alicia, I have Andrew, I have Jess. So do you have any questions on that? I'll take silence as a no. Okay. So just over at the top of the page, it just has a little table for the different parts. So are we happy to move?
Yep.
Okay, so click to the end. So just in terms of what they are. So the extene is just a really thick outer coat. This is the thing if you're allergic to pollen, it's that outer coat is what's annoying the crap out of you. Intene is just a little thin inner coat. And then you have the 2 nuclei. So the jobs of them are very specific. The tube nucleus, it's going to make a tube down your stigma, and it falls apart or dies just when it gets to the ovary. So this will make a little bit more sense when we do fertilization. So until we get to there, it won't make sense. And the generative nucleus will make your male gametes. So we haven't made the male gametes just yet. And I think at the end of page 176, it says not quite male gametes yet. So if you want to highlight that part for yourself, because that is important. When we do the girl version, it makes you have your female gametes are made once you go at the end of this process, but for the males, it's not until it's in the girl bits do you make your male gametes. So the part that they're going to love to ask you if they ask is, and it's usually the male version they like to ask, is outline the process of male gamete formation. So it will have this beginning and it might have a little extra bit for the end, but it'll have the microspore mother cell, the meiosis, tetrad of pollen, 4 haploid cells, and undergo mitosis to make 2 nuclei. Okay, so there's 2 QR codes. There's on page 176 a detail of formation, and on page 177 there's how to revise for them. So I have my screen big, but I can't see everybody. So I can see Andrew's head and its Flo. So who is not my list? Andrew, Clara, Flo, Hannah, Jazz. Who's John? Somebody's on John's iPhone. Okay, that's all right. I was trying to work out. Am I going mad? So Laura, Robin, Bannon. Yeah, that's everyone. Okay, happy to move? Yeah, thank you. So again, just to highlight this, we haven't made male gametes yet. We're really, really close, but we haven't made them. So that I think is on the bottom of page 176 for you. So then just the last bit of this, and you don't have the picture, but you have the words. So when the pollen grains have matured, the walls of your anther dry and split, and your pollen grains get ready to be released for pollination. Okay, happy to move? Alrighty. So we are at this. Okay, so we'll get this done really quickly, and then you can ask some questions. So these are the girl bits as such. Unfortunately, I bought roses. I usually buy lilies, because you can't really see the girl bits in them. You have to get really into them to see them. Because I usually buy lilies. So what do I want? Change my screen, stop my share. Okay, so where is me? I'm up here. So if you have flowers at home, you can have a look. Can you see when you see sideways at the bits that are yellow here, you can see one little bit sticking up higher than everything else. So that little teeny tiny bit, that is the girl bits, and all these bits over here, these yellow bits out here, I get this right. These are all the anthers. So there's way more, loads and loads of anthers, and there's only one stigma, and that is going to be the tallest bit always in the flower. So like when I teach junior cycle, I tell them, you should always be disgusted when you look at flowers, because they're literally having sex in front of your eyes. So the tall bit is the stigma, and you now will have the stigma here. So this is this tall bit that's sticking up here. So just in terms of the parts, we've done them already, but you have your stigma, you have your style, and you have the ovary. So they were the bits that we labeled earlier in the day. And then we just start looking at what's inside them. So inside an ovary, you will have many ovules. So the ovary is like the bulge, and in there will be many ovules, and the ovules has this wall down here, and they're called integuments. This little part number 6 is a little hole, and it's called a micropyle. And because you're going to have, it's like the tapetum in the anther, you're going to need an energy store. You have a thing, it's called a nucellus. Now, in reality, our memory serves, this has never been asked. So it's whether or not they will, I don't know, but it has never been asked, these little parts. So similarly with the anther, so the picture with the tapetum, and what else was in there? The fibrous layer, that also has never been asked. So just to say, every ovary will have one or more ovules depending on the type of flower. Each ovule will have 2 walls, and these are called integuments. And between them is this small opening, which is the micropyle. And what's going to happen when we do fertilization is a pollen grain is going to land up here on top of your stigma. Your tube nucleus literally burrows its way all the way down here, and it's going to go into your nucleus in through your micropyle, because that's the little opening. And as we said, then the tube nucleus degenerates, it literally falls apart here, and our generative is going to have followed it all the way down, and it's going to make the male gametes along here. But we'll come to that when we do fertilization. And then the nucellus is nutrients, energy source. So this hasn't been asked, and the cross section of your anther has not been asked. It's always been the blurb for the male and female gamete formation. So I can only see some of you, but happy to move. Okay. Alrighty, so again, said the pictures may help you. It is the blurb that they're more than likely going to look for. So you can see this time you have an extra bit around your cell. So this, it may help you to then kind of do some visualizing. So when we called the male starter was a microspore mother cell, this one is a macrospore mother cell, because technically it's bigger. And again, it's diploid 2N. So this round bit up here is your nucellus. It's kind of like the food store or the energy store, because this nucleus, I'm either making new cells or I'm making new nuclei. So last time our microspore mother cell, what was the process it did? If you think it's mitosis, point to your left. If you think it's meiosis, point to your right. And hopefully you'll all point. Okay, so that side is my left. So that's the mitosis side. This is the meiosis side. Okay, so thank you, Alicia. Thank you, Laura. Okay, so we're not sure if we're all going the right way. Okay, so it's going to undergo meiosis. So it does the exact same thing. It's a different name because it's a macrospore, but it undergoes meiosis. And how many cells did I make last time from meiosis? Thank you, Robin. Okay, you make 4 cells. So this time the 4 cells aren't stuck together as they were, but I make 4 haploid cells inside here. And here is where females were way more complicated. So even in female reproduction, every month we generate loads of eggs ready for ovulation, but then we get rid of loads of them and we only ever use 1. And similarly here. So while there are 4 haploid cells, 3 of them disintegrate. So while they make 4, 3 fall apart or just are broken down, they're not used, and only 1 is going to be taken forward. So in your book, I have drawn like an arrow down to the next row, and I've put in, you have the picture of the cell that you have then. So it's 1 cell is this happens to. Okay, it's going to do that because it's going to get in the way. Why are you not letting me do that now? Nope. Okay, sorry, gotta go out to come in. Discard. Interview. Okay, so one of those cells is going to take forward and it's now going to undergo. So in your book, you will have a picture of a cell. You will have an arrow. So this is, you're going to put mitosis. You're going to put 3 times. And it's the nucleus. So it's like last time with the male version. It's the nucleus that undergoes mitosis. So the first level of mitosis, 1 nucleus becomes 2. In the second version, 2 become 4, and the third time 4 become 8. So you're going to have 8 haploid nuclei in here. The colors aren't hugely important other than 5 of them are going to disintegrate, and they will happen to be the 5 that are drawn in black. So if you want to put X's, so you make 8 nuclei, but 5 of them disintegrate. And this is what you end up with then. So you have an egg nucleus, which is haploid, and you have what are called 2 polar nuclei. So these are these white ones. And these are your female gametes. So this is the female gamete. But this, the girl gametes are now ready to go. We haven't quite made the boys ones. See a time. So are you as happy as you can be? Do we have any questions? So I'm going to move forward unless somebody says no. So you don't have this. I'll send it as a Word document if you want to print it, but I think this may help you to see what's similar and what's different. So if you want to have a go at doing this now, and what we can do is I can fill it in for you and I will send it to you then when it's filled in, or you will have it filled in yourself. But this is the last bit we're going to do today. It's part of your homework, so we can get it done together. So if you wish to, I am unmuted. I can mute me and you can unmute yourselves and you can talk it out or not, but it may help you. So it's looking for what's the initial cell name, what's the type of cell division in your first step, what do you make in your first step, what happens to those cells, what's the type of division in your second step, what's the number of times the nucleus divides, what do you make in your second step, and then what are those nuclei, so what are the names. So you can either decide to do it yourselves and we can check in, or you could put on your mics and you could talk to each other. Imagine that. You could talk to each other. So I'm going to I'll mute me and I'll give you 5 minutes and then we'll check through. So if you want to put in chat when you're done, then just go through it very quickly. So we're waiting on 4. I think we're just waiting on 1, and that's everyone. Okay, so we're still sharing and you can see. So I am hoping that the initial cell name, so Robin, initial cell name from male gamete formation. I'm going to make everyone turn on their mics now at least once.
I said microspore.
Correct. So this is going to be the microspore mother cell. So this is the diploid, the 2N. So who's next to her? Jazz, you're next by my pictures. So what was the type of cell division for the first stage?
Meiosis.
Yep. Okay, Velen, you're the next one. What did you make in the end of your first stage for the male?
4 haploid cells.
Yeah, so don't forget to put in the tetrad. So the tetrad, and that is 4 haploid cells. Okay, what happens to those cells? Hannah, you're up next.
They separate.
Yeah, and then, yeah, they separate. So that's this. And it's each cell is going to go into the second step. So just be aware it's that. Okay, who is next to my lovely pictures? Mark, it's you. So then the type of cell division in your second step?
Yeah, mitosis.
Mitosis. And Laura, you're up next. The number of times the nucleus divides.
2.
So how many nuclei do I end up with after this?
Hold on, let me check. 4.
So no, so we're at the very kind of bottom row.
Oh, 2.
So I end up with 2 nuclei from 1. So how many sets of mitosis have I done?
2.
So if
I'm confused.
if you do mitosis, 1 cell becomes 2 if I did it once, and if I was to do it a second time, 2 nuclei would become 4.
Okay.
So it only does mitosis once.
Okay.
Okay, and then our second step, Zara, this is you. What do I make? So what are the names of the nuclei? You have to unmute, I think.
Oh, 2 haploid nuclei,
Yeah, but what are they called?
generative nucleus.
Yeah, so we're going to have a generative nucleus. And what's the name of the other one?
Tube nucleus.
And tube.
No.
So yeah, so tube nuclei, and I don't have room in mine, but they are both haploid. And then what happens to these nuclei? They are going to be incorporated into your pollen grain. Okay, I need to see now who's next in my bed. So Clara, you're up. Female gametes, what's the name of the initial cell?
I said microspore mother cell for that one. I
So,
got the mixed up.
okay, so remember we're saying sperm, teeny tiny, micro, and then eggs for females are huge. So this is going to be the macro. So macrospore mother cell, and again, it's going to be diploid. So then our type of cell division, Alicia?
Meiosis.
It is. So meiosis. My products in my first step, Andrew, what do I make after this meiosis?
I said 4 haploid cells.
Yeah. So 4 haploid cells. And what happens to these now? Flo, can you hear us? See, Flo now could be having a snooze because her camera's on.
No. 3 disintegrate.
Yeah. So 3 disintegrate and 1 move taken on, or 1 to step 2. Thank you, Flo. Okay, so are we back to Robin? We're back to you. Type of cell division in your second step?
Mitosis.
Again, okay, so mitosis. And where am I? So Jazz, number of times the nucleus divides this time.
Once.
So how many nuclei do I end up with in the girl version? 1, I start with 1, and when it goes through mitosis, how many do I end up with?
4.
So,
Oh, sorry. 1.
so if you look at the bottom of page 178,
Yeah.
so we have that 1 cell in that bottom row, or 1 nuclei. How many nuclei then? How many balls are in the next picture?
8.
8. So if 1 nucleus becomes 2 for the first round of mitosis, then 2 become 4 for a second round of mitosis, 4 becomes 8. So there must be 3 rounds of mitosis.
Yeah.
So there's 3, and then our product is, Hannah?
8 haploid nuclei.
Yeah. And what happens to these nuclei?
They disintegrate. Well, 5 disintegrate.
Yeah, 5 disintegrate, and then what am I left with?
You're left with 3. And I don't know what they do.
Okay, so we haven't done what they do, it's just what they are. So we get 3 that remain, and we have an egg, 1 egg nucleus. So I'm not going to fit that in, I don't think. So 1 egg nucleus, and had I not given you that part? Had we named them? Had we named them? Yeah, we had named them. So I have 1 egg nucleus, and I have 2 polar nuclei, they're called. And they're all haploid. So if you have your little highlighter pens, so the things that are the same are they undergo meiosis. Meiosis makes 4 haploid cells. They both undergo mitosis. So you have meiosis, 4 haploid cells, mitosis. I'm going to change my color pen, go to green, and then just the difference would be in the micro versus the macrospore. It would be your tetrad makes it different from your boys versus your girls. The fact that we're getting things that disintegrate. mitosis once, mitosis 3 times, and that we have 5 disintegrate. So there are some things that are similar. So we have the idea of the names of the nuclei are different, and that you're getting disintegrations for the female that you're not getting in the male. I don't know whether that will help. Seeing them side by side. So when we say goodbye, what I will do is the recording has to convert. I will put the recording onto YouTube, and in the homework assignment, I'll put the YouTube link in. So if you want to go back and look at it, you can. I've removed that life plus question. So essentially the question that's in there is, they nearly always ask the question on the male gametes. It's not on the female, but I've put in what would you write for the version of the female. If they ask you the female question, what would you write? So there will be a class notebook page for you to hopefully put back everything in. I am going to spend this evening going through notebook, because I've been physically marking papers other people did before Christmas break. Can I stop my share so we can see your lovely faces? Tomorrow I will put up in the morning just a little Google form. So it's a bit on the learning you did today, and it's going to have a little bit on what we're going to do tomorrow, just to test you, because you do know it. It's on different types of pollination, but you actually will know it if you just take the little time to think. And then we'll do the tiny little bit on pollination, and we'll see where we'll go. So I have no idea what next week is going to work like. Are we going to be in school? Are we not? If you're in school, are you doing like option 1 line, a couple of hours of that at a go? Are we following a timetable? We're just going to have to suck it and see. Hannah, your eyes are looking exceptionally blue. So they are. They look lovely.
Mine.
Yeah, whatever way the light is shining in.
Oh, thank you.
Beautiful blue eyes.
I think it's a trick.
It's a good trick. So you really have one of those lighting bars, don't you? You've seen the ones that all the people on TV, they have a lighting bar, so they're all,
It's just a reflection.
so they're all gorgeous.
Snow.
All right, so lads, we hope to see you in school, all going well, and if not, we'll see how we go. We're all happy as we can be.
If we're back next week, do we have the revision test that we were supposed to have?
Irrespective, we will hit that one. So that just gives you your last chance to make sure you've seen everything we have done prior to mock. We'll wait and see, because we've no idea what's going to happen. But at the moment, we'll take it that we're having it, and if it changes, it changes. Is that okay?
Yep.
Alrighty.
Thanks.
So look, be safe, guys. It's lovely to see your faces. And we will talk tomorrow. So I'll put the Google form or the Microsoft form up for the morning. It'll be up at 8:40 in the morning, and you can have a go at it, and then we'll do our little bit of our lesson. Okay, if you're talking to any of the others that haven't been here, so we have 11 out of, we've 7 people missing. So I know Millie was the one person that came and said, I have to collect my books. But if you're in contact with any of the others, let them know. Alrighty, so happy Thursday. Be safe. Bye.